Okay, so this next immune series, is going to be on antibodies and antigen, and this is sort of a series before I start talking about B cells. So the first thing we have to discuss are the concepts of antigen and antibody. Antibodies are pretty straightforward. Antibodies are proteins that are made by your B cells. So they are encoded in your DNA, and they are produced and released in response to a stimulus by a B cell. So these are made by B cells. Now, what's important about antibodies is that the antibody will bind to antigen. And so the definition of an antigen is a molecule that antibody binds to. Okay, so this is the definition of an antigen, a molecule that antibody binds to. So what does a molecule mean, and where does a molecule come from? Well, the molecule can be viral, bacteria, other foreign, and these would all lead to an infection, and the antibodies would help protect. Okay, so that's typically what antibodies do. Antibodies are going to bind to antigens. Most of the antigens are going to be foreign antigens from some sort of pathogen, and then the antibodies are going to help to eliminate the pathogen and protect from the actual disease. And then an antigen can also be self. And an example is diabetes type 1, where you have a self-antigen that stimulates the immune response to attack and kill your pancreatic beta cells. Others are rheumatoid arthritis is a type of self-antigen, and in this case, the antibody is binding self, and this is autoimmunity. Okay, and so protection is, of course, what we want. Autoimmunity is what you don't want. Now, just a little bit about an antibody and its structure. The antibody is typically drawn as a Y. So you have what are called light chains and heavy chains. So I'm just going to change the color of my heavy chain. So the heavy chain is going to be in purple, and your light chain is going to be in black. So you have two heavy chains and two light chains. And this is a typical IgG molecule, and it could also be an IgE molecule. IgA and IgM look different, and we'll talk about those more when we talk about B cells. But this is just what an antibody looks like. Now, what's important about the antibody is its function, of course, because it's going to protect or cause autoimmunity, and it has to do this by binding the antigen. So the antigen binds at the point where the heavy and light chain meet at the ends of the Y. So these are the antigens. Okay, and so there are these blue circles that this antibody binds to. And typically it's going to be a foreign antigen. It's going to be from a virus, or it's going to be from a bacteria, or something along those lines. Now, binding of the antigen is a function of the molecule, the antibody molecule, but it is not what we call an effector function, so it doesn't do anything. The antibody just binds to the antigen. What actually does something is the trunk of the antibody, so this area right here, and we call this area Fc. And the reason it's called fragment C is because it's going to bind to Fc receptors and provide a function to the antibody. So this is where the Fc portion is where the antigen functions. And we're going to talk about different functions of different antibodies depending on their subtypes. We know that IgE is the antibody for allergic responses, so we'll talk about what that Fc region is actually doing with respect to the antibody function of the IgE molecule. And then we'll talk about the other subtypes as well. So you have your antigens binding, and this area where they're binding is called the Fab region, because it's the fragment where the antibody is binding to antigens. And the functional region is the Fc region, and that's where the actual antibody molecule will do something once the antigen is actually bound. All right, so let's just put this into a little bit more context and give an example. And also, this is a very good time to talk about specificity. So we have antibodies, and they're going to have a heavy and light chain. And at the end of these antibodies, they're going to be able to bind to a specific antigen. So we have an antigen here that is a triangle. And so the end of the antibody here would look like a pocket of a triangle. And it'll allow that red triangle to fit inside of the pocket. If you had that blue circle would not be able to fit inside of that triangle pocket of the antibody binding site. On the other hand, on antibodies binding to the circle, you'd have more of a circle-looking antibody. And so that circle-looking antibody would now be able to bind those circle antigens. And so this brings up specificity. So every antibody can only bind to a single antigen. Now I'm drawing them as circles and triangles, and of course that's not how proteins look. Proteins or carbohydrates have chains, they're folded, they have dimensional structures to them. So the antibody will bind to one portion of that. So to put this into a little bit more context, let's pretend we have a bacteria, and our bacteria have a flagella. So here's our bacteria, and this is their flagella. Now the bacteria flagella will have different regions. So let's say that this region up here has these little triangle spikes, and this region over here has these little round spikes, and then we'll have another region that has square spikes. What I'm trying to explain here is that for every antigen, in this case it's a flagella, you'll have multiple binding sites, but only an antibody that's specific for the triangle will bind to the triangle site, and only an antibody that is specific for the circle will bind to the circle site. And that's how it is for all of them. And then you'll have another one that would bind the square. And that one will be specific for a square. So even though you have multiple antibodies binding to this bacteria, they're all binding to different antigen sites on the bacteria because they're very specific. A triangle binding antibody will never bind the circle portion, and a square can't bind a triangle, etc. So that's basically how antigens and antibodies work. Many different organisms have hundreds of thousands of different antigens, and so you're going to have hundreds to thousands of different antibodies made during an infection. There will be dominant antigens, and when we start talking about infections, we'll talk about dominance in actual bacteria and viral antigens, and how that plays a role in the antibody that are produced during an infection. So that is the end of this mini-series.